work was really this work was really a huge team effort between many different scientists and was started by a prior postdoc in the lab, Esther Florsheim, who's co-author with me on the manuscript. So all of the data within is both of ours, not just mine. So what I'll be talking to you about, the way that I've laid out this presentation, I'll start off with some background, trying to frame the problem that we're approaching. Then I'll talk about the goals of the project, dive into data where I'll talk to you about this aversive behavior that we studied throughout the manuscript, talk about what we think the mechanism of this avoidance might be, and then summarize the findings that we've discussed, and then talk about kind of what the future of this area we think at least looks like, and open it up for discussion. And because I like a roadmap before I get started, I wanted to just show you kind of a summary of the findings that I'll be presenting. So what we think that we found is that sensitized mice, when they ingest allergen, this binds to mast cells via IgE Fc epsilon R1 immune complexes. And we find that these mast cells are able to produce a number of different mediators, but of these, we find leukotrienes are crucial in driving their ability to make a choice to not drink the allergen in question. How this is happening downstream of leukotrienes is a little bit less clear, but we identify that epithelial production of a hormone called GDF15 is at least partially responsible for this behavior, although likely direct neuronal routes contribute as well. So I want to broaden this discussion by first talking about our senses in general. So one of the things that's amazed me for a lot of my life is that we're able to, for instance, sense delicious cookies being made in the oven. And when we bite into them, we're able to perceive that they are sweet, they're salty, they have fat, and that there's something that is intrinsically rewarding about them. And similarly, we're able to listen to different music and be able to perceive different pitches as well as amplitudes of said sound. And these various songs can really take us back and lead us to certain periods of time in our life and kind of elicit certain emotions as well. However, while we normally consider these specialized senses, such as taste, olfaction, as well as hearing, to be realms of neuroscience, neurons really can't do the job alone. And so I would like to remind you that there are a variety of specialized cells that are co-opted by neurons to expand their sensory repertoire to better perform their function. For instance, we know that on the tongue, there are taste buds, which are composed of specialized epithelial cells that are topographically innervated by different sensory neurons. And these specialized epithelial cells express molecular repertoires that enable them to very exquisitely sense different tastings, such as bitter, sweet, sour, umami, and salt. Similarly, in the inner ear, we have specialized epithelial cells called hair cells that, based on the bending of the cilium that they expressed, we can detect different pitches and amplitudes of sound. And what I'd like to convince you of throughout this presentation is that the immune system can be thought of as an extension of the sensory repertoire of the nervous system. And in the case of the specific project I'll be talking about today, the mast cell expands the sensory repertoire of the nervous system near infinitely through its ability to bind to antibodies, specifically IgE. And these interactions between mast cells and neurons, or immune cells and neurons more broadly, are capable of eliciting a wide variety of defensive behavioral reflexes. And these reflexes that are initiated depend not only on the type of inflammation that initiates these circuits, but also where these circuits are located. For instance, respiratory viruses that proliferate in the nasal mucosa will trigger the release of perhaps prostaglandins that, through actions on local neurons there, might elicit sneezing. whereas bacterial pneumonia is found in the deeper airways, may elicit other reflexes such as cough. And we believe that food allergies, uh, and specifically food allergens by binding to mast cells, will elicit a separate set of behavioral reflexes, including avoidance behavior, but of course not limited to it. And the reason that we're interested in studying how allergen recognition by mast cells is capable of eliciting this avoidant behavior is really twofold. One is that we might learn something new about how the immune system is capable of driving negative valence signals, such as malaise, abdominal pain, and bloating. And the other is because we think that perhaps this ability to drive avoidant behavior is one of the reasons why allergies might exist in the first place. And kind of the rationale for thinking about allergies in this way is through drawing a comparison with another phenomenon that is studied in behavioral neuroscience called conditioned taste aversion. And we all can relate to this phenomenon if we've ever had food poisoning, whereby the ingestion of certain noxious food substances is sensed by our taste buds, our smell, and that these various sensory cues are paired with some sort of adverse effect that the toxin drives. For instance, if we've had food poisoning from a sandwich that has been out for too long, our nervous system does not know directly what had the toxin, but makes an inference that the smell or the taste of food that temporally associated with said negative feelings from the toxin was the cause. And of course, then maybe that same sandwich will become much less palatable to us for the following days, months, or even years. In much the same way, allergic sensitization occurs not through directly targeting uh, the food item itself, but rather is because the noxious substance that triggered some sort of adverse effect to the epithelial cell, for instance, IL-33 release, is found in the same temporal and spatial localization as food antigens that are ingested at the same time. And we believe that this immunological pairing of noxious substance-induced adverse effects with food antigens elicits type 2 immune memory, of course, but one of the consequences of that immune memory is not only epithelial remodeling as well as peristalsis and kind of things we normally think of when we think of allergic responses, but it also enables a unique complementary type of long-term avoidance behavior to that elicited by conditioned flavor aversion, for instance. And the focus of the talk today and of the paper was really to try to understand how this conditioned aversion that occurs in the context of food allergies actually happens. We're not the first people to have described or studied this phenomenon, and I want to give a shout out to many groups that have studied this before us. The first well-known group that we could find was this group in Brazil led by Kara et al., which described that allergic sensitization of mice against a given food protein of albumin, which is a protein found in egg white, elicited avoidance behavior to that specific food protein when administered in the drinking water, and that this aversion was highly antigen specific because the mice were able to distinguish between BSA and ovalbumin. The next experiments were super interesting and almost sci-fi-esque. This group found that you could actually hyperimmunize mice using Freud's adjuvant and then transfer the serum of these immunized mice into naive recipient mice. And then these naive mice would develop the same specific antigen avoidant behavior to the initial immunization, suggesting that some factor in the serum is actually critical for driving this avoidant behavior. And finally, a few years later in the early 2000s, a group at University of Sao Paulo using antibody-mediated inhibition of IgE found that the factor in the serum likely responsible for this behavior was likely IgE antibodies. And I'm not sure if everyone in the audience knows how allergic immunity works, so I'll give a very, very brief refresher, oversimplified, of course, for the purposes of the talk. So we know that IgE antibodies develop when T follicular helper cells within the draining lymph node produce IL-4 and cause B cells to class switch to be able to produce the IgE isotype of antibody. And IgE antibodies are highly specific for the allergen of interest, and they circulate at very low levels in the circulation, but they bind with extremely high affinity to localized cells within the tissue and in circulation, namely mast cells and basophils. And they're capable of binding to these cells because these cells express the high affinity receptor for IgE, Fc epsilon R1. And once loaded with these antigen-specific IgE complexes, when a low dose of allergen is encountered, mast cells or basophils can rapidly degranulate, leading to a wide variety of changes within the tissue, ranging from vascular changes to changes in mucus production by the epithelium, and of course, of interest today is changes in behavior through actions on neurons. The primary questions that we sought to answer in this publication were to better characterize allergen aversion and define exactly what was going on at this time span. We wanted to work out the mechanism and confirm or refute the role of IgE Fc epsilon R1 signaling in this phenomenon. If we could define a role for this, we wanted to make sure that we could tell whether mast cells or basophils were responsible, and define the mediators that these mast cells or basophils were producing that might contribute to this behavior and what neuronal substrates that it plugs into. The way that we ended up approaching this phenomenon was very similar to these initial Brazilian papers that I described. And it was established in our lab by Dr. Esther Florsheim, who's now an assistant professor at Arizona State University. We take bulb C mice and inject them subcutaneously with either ova paired with alum, which is a type 2 skewing adjuvant, or an antigen-free control. We do this 2 immunizations 1 week apart, and then 1 week following the second immunization, we acclimate them to 2 bottle preference test cages, and then give them a choice between drinking egg solution or water. And we utilize cages where we're able to detect the number of licks that mice take of either bottle over time, and we're able to then form preference indexed for allergen between different sensitizations or different genotypes of mice. When you do this in bulb C mice, what you find is that upon first allergen exposure, and this is plotting the cumulative licks of allergen over time, that sensitized mice very rapidly diverge from their non-immunized counterparts, showing minimal consumption of ova, whereas the non-sensitized mice can continue this consumption over time. And you're able to plot the preference index for ova in the span of 4 hours and observe that at both the first and second day of preference testing, these bulb C mice strongly avoid allergen solution. So what's going on with these mice that's capable of driving this shift and this decision not to consume the allergen? Well, if we look in the tissue of these mice, so if we isolate the small intestine of these mice into lamina propria and epithelial fractions digested and stained via flow cytometry to measure changes in immune cell populations, we see a whole lot of nothing at the time where this behavior occurs. We're not able to detect significant differences in the number of mast cells, nor dendritic cells or macrophages in the small intestines of these mice at this time point. And moreover, we're not able to detect differences in monocytes or neutrophils. And although we have a mild increase in eosinophils, this is very small. And at this same time, so there's really not robust intestinal inflammation happening in these mice. And when I tell you about food allergy, most people think of symptoms such as diarrhea and anaphylaxis, but at this time point when this behavior occurs, for instance, if you give a single dose of allergen to sensitized mice shown in blue, you do not detect a significant decrease in GI transit time whereas you do if there's repeated exposure to allergen leading to diarrhea. So this behavior can happen extremely early when really no other major signs of a food allergic reaction are occurring. Now, one thing that we did observe at this early time point, however, was neuronal excitation in select brain regions. And one of the most well-described brain regions that's elicited by numerous different negative externalities, including pain, itch, and various nausea-related pathways that can halt food intake, is this NTS to lateral parabrachial nucleus to central amygdala pathway. And we observe that in sensitized versus control mice, given a single intragastric gavage of ovalbumin, the same gavage between these two mice, we observe a robust CFOS activation in the NTS, lateral external parabrachial nucleus, and central amygdala. And as you can see here, via the quantification, this was highly significant. And so we think that these findings tell us that food allergies can manifest in ways different from how we would typically expect and might simply be able to elicit changes in food preference before much more severe symptoms that we traditionally think of. With this in mind, we began investigating how this avoidance behavior is actually initiated. How is it that the allergen in the gut is being sensed, and how is this being relayed to the nervous system? So using mice genetically deficient in IgE antibodies, which we confirmed via serum ELISA, shown here, we're able to see that IgE deficient mice that are sensitized to ova fail to reduce the number of licks of ova during a two-bottle preference test, and they actually display no difference as compared to non-sensitized control mice with regards to their ova preference, whereas their wild-type counterparts still develop this robust avoidant behavior. And we've done similar experiments that I don't have time to get into today, showing that Fc epsilon R1 is also required for this behavior, and that IL-4 receptor alpha, which is required to produce IgE, is also necessary for this behavior to occur. So we next wanted to tackle the question of whether mast cells or basophils was required for this behavior. And these are the two cell types in mice that express the high affinity receptor for IgE. And I wanted to briefly go over some differences between these cell types. So despite mast cells and basophils expressing a lot of similar things, including the ability to produce histamine in various eicosanoid species, mast cells are thought to be predominantly tissue resident, whereas basophils are circulatory. And there are two types of mast cells. Connective tissue mast cells are seated during development, and mucosal mast cells in response to allergic inflammation, infiltrate into tissues and become more localized to epithelial cells, as is shown here. And so really mast cells can be predominantly thought of overgeneralization as tissue intrinsic targets of IgE, whereas basophils may be able to be considered more systemic targets. And important for the experiment that I'll be talking to you about on the next slide, the repopulation kinetics of these cells differs dramatically upon depletion, whereas mast cells, it takes them 1 to 2 months before they come back into tissues, whereas basophils take only 1 or 2 weeks before they come back into blood. And so we utilize a mouse that is called an RMB mouse, or a red mast cell basophil mouse, which expresses TD tomato and human diphtheria toxin receptor under the Fc epsilon R1 beta chain. And this allowed us to conditionally deplete both mast cells and basophils. And then we're able to wait afterwards, prior to performing the two-bottle preference test, and we were able to find a window where mast cells shown here, either in the peritoneum or small intestine, are significantly depleted, whereas basophils found in the blood have come back. And this has allowed us to directly test a role for mast cells in driving this avoidant behavior to ova. What we found was that these mice consumed way more allergen solution as compared to wild-type mice, and similar to the IgE knockouts, didn't display any difference in ova preference, whether they were sensitized or not, suggesting that mast cells are essential for driving this avoidance behavior. Which that was really interesting, but the next rational question became, what are these mast cells actually producing to drive this behavior? And this became a complicated question. So mast cells and basophils are packed with a ton of different substances, and moreover, they can produce other potent mediators on different timescales. And so the way that we really approached this problem was to broadly group the different types of mediators based on the timescales that they're produced under, and then look at the behavior of mast cell deficient versus sufficient mice and see where they diverge. And when we did this, if you compare mast cell deficient sensitized mice to wild type mice, and you plot their preference for ova on the y-axis over time on the x-axis, what you observe is that the wild type mice and mast cell deficient mice diverge from one another extremely quickly in the matter of seconds to minutes. And so we initially kind of thought that, at least for the initiation of this behavior, cytokines and chemokine production by mast cells would likely be too slow to be able to account for the change observed. And then after this, Esther and I, over many, many experiments, worked through a lot of the at least most well-known mediators that are produced by mast cells, either using pharmacological or genetic means, and found that the vast majority of these substances were actually non-essential for the avoidance and did not alter ova preference in the slightest. One class of substances that we did get a hit on, so to speak, was leukotrienes. Leukotrienes are derived from membrane phospholipids. They're a class of eicosanoids, and they're generated through the conversion of arachidonic acid to leukotriene A4 by an enzyme called ALOX5. Leukotriene A4 can then be converted into LTB4, which is mainly well known to be involved in cellular chemotaxis, or through LTC4 can be converted into LTC, D, and E4, which are collectively termed the cystinyl leukotrienes. And these cystinyl leukotrienes have kind of gained popularity in recent years and are well-known mediators of asthma, but have also gained notoriety recently due to their profound roles in itch. And at the highest level, these mediators can also mediate things such as bronchoconstriction, as well as vasodilation, and in excess can lead to anaphylaxis. So we began, because leukotrienes had not been characterized ever before in food allergies, at least in mouse models that we could find, we first just looked to see if transcripts for ALOX5 were induced in the intestines of food allergic mice relative to non-food allergic mice, and whether this depended on IgE and mast cells. We indeed found this to be the case. We found that wild-type sensitized mice had about a 2 to 3-fold induction of ALOX5 as compared to non-sensitized mice, and that this was entirely dependent on IgE and mast cells. Moreover, we're able to analyze a previously published single-cell RNA-seq dataset that used a very similar food allergy protocol to the one that we utilized, and we found that really mast cells, but not really any other immune cells, expressed all of the enzymes necessary to produce cystinyl leukotrienes within the food allergic intestines. And so this tells us that mast cells in the intestines of these mice likely can produce leukotrienes, but are the leukotrienes actually required for the behavior observed? To test this, we started off by just treating these mice 1 hour prior to the 2-bottle preference test with Silutin, which is an antagonist of ALOX5. And this led to the Silutin-treated group, shown in red, consuming significantly more ova over the course of the 4-hour trial relative to the vehicle-treated group, shown in black. And this led to at least a partial effect on the magnitude of this ova preference on the second day of testing, which is shown here, whereas on the first day of the preference testing, the ova consumption was actually not significantly different from non-sensitized mice. So this suggests that leukotrienes in general are required for this avoidance behavior. However, we wanted to validate this using genetic knockout models. We used mice that were genetically deficient in LTC4 synthase, which is necessary for cysteine production. And we confirmed that these mice indeed lacked the production of cysteine by measuring the quantities of these leukotrienes in the duodenal tissue of food allergic mice that were acutely challenged with allergen. And you can see that the wild type sensitized mice had a very strong, about 20-fold induction of LTE4, which is the terminal cysteine detectable in the duodenal tissue relative to non-sensitized mice, and that this was entirely dependent on LTC4 synthase, as the knockout mice did not display any quantity of this. And then subjecting these same mice to the two-bottle preference test paradigm that I've been describing to you revealed that the LTC4 synthase deficient mice did not display any decrease in OVA preference relative to control non-sensitized mice, suggesting that within leukotrienes, cysteine leukotrienes are likely the class of mediators that are required for this behavior. What's more, we found that administration of LTC4, but not D4 or E4 intragastrically to mice was capable of decreasing OVA preference, and these mice were naive. So we literally just took wild type bulb C mice that were not allergic, gave them varying doses of LTC4 via intragastric gavage, then gave them a preference between OVA or water, and LTC4 administered in this way was capable of decreasing preference for OVA, whereas LTD4 and E4 were not. And since LTC4 binds preferentially to cis-LTR2, we also observed that cis-LTR2 deficient mice, shown in red here, displayed an amelioration of this allergen-induced avoidant behavior. So in summary of this part so far, we found that IgE-mediated mast cell activation likely triggers the production of intestinal cysteine and in particular, LTC4 produced by these mice acting through cis-LTR2 is required for this avoidant behavior. People might be wondering, do these leukotrienes affect the magnitude of the humoral response to allergen or the degree of mast cell degranulation in response to allergen? We've measured this, and at this early time point where we performed the two-bottle preference test paradigms, we were unable to detect any difference between OVA-specific IgE between mice that can produce these cysteine leukotrienes or not. Additionally, mast cell protease levels were not significantly different between these two strains upon allergen challenge, suggesting that these leukotrienes aren't just preventing mast cells from working, but are likely actually produced by the mast cells to directly drive this behavior. Okay, and the next question that is obvious is we wanted to understand how these leukotrienes were being perceived by the nervous system in order to change the behavior of these mice. And there are really three main pathways, at least three of the most well-known pathways that could mediate this sort of a change in behavior. And that's through the activation of TRPV1 positive nociceptors or proreceptors. These neurons have been shown to be able to mediate itch in response to leukotrienes by Brian Kim's group, Mark Hoon's group, as well as Isaac Chiu and Caroline Sokol's group. So these were very reasonable pathways that could be mediating the effects observed. Moreover, the vagus nerve is a conduit between the gut and the brain that might be able to measure feelings such as satiety as well as nausea. And finally, we considered a humoral route whereby substances released by mast cells directly or indirectly might be able to make their way to the brainstem and drive a similar change in behavior. To test the role for TRPV1 positive neurons in this behavior, we had performed experiments with racinoferatoxin mediated depletion of TRPV1 positive neurons. However, these did not make their way into the manuscript because we observed that RTX administration to wild-type mice that were non-allergic actually in and of itself led to the mice not liking ova very much. And so to get a little bit more clarity on whether these neurons were required for this behavior, and this is very preliminary, so take it with a grain of salt, I've backcrossed TRPV1 DTR mice onto the bulb C background strain and genetically ablated these neurons by administration of diphtheria toxin. And then because different neurons might sense different levels of allergen and levels of leukotrienes in the intestines, I employed a behavioral paradigm that we adopted from Marcelo Dietrich's lab, who's a neuroscientist that we collaborate with. This is basically the exact same two-bottle preference test paradigm that I've been describing throughout this talk, except every week the dose of ova in the drinking water is escalated such that the first week the mice are given a choice between water and 0.1% ova. Next week it's water and 0.3% ova, then 1% ova, then 3% ova, etc. And we thought that this was important because it could be these neurons are playing a role only at, for instance, low thresholds of allergen. However, our preliminary analysis of these mice with sensitized wild-type mice shown in red and TRPV1 ablated mice on the right, wild-type mice on the left, reveals that the TRPV1 DTR mice really do not have any difference in the development of this avoidance behavior to ova across any of the doses of allergen tested. And so we think that these neurons may not be required for the avoidance behavior. However, we do observe, and this is preliminary, perhaps a slight difference upon low dose over re-exposure on the 5th week of testing. So maybe these neurons contribute to the development of enhanced sensitivity to allergen, for instance. With in mind that TRPV1 depletion didn't significantly affect the avoidance behavior, we wanted to test a role for the vagus nerve in mediating this. And so with Marcelo's lab, and specifically with experiments performed by Madhya Godesgar from his lab, we conducted subdiaphragmatic vagotomy of previously sensitized mice before two-bottle preference testing. Madhya was able to confirm that this vagotomy was successful by lack of staining for fluorogold upon IP administration of fluorogold being found in the nucleus of the Tractus solitarius, which is found right here. This just confirms that the vagotomy was successful. And the results from this experiment really showed that both sham-operated and vagotomized sensitized mice both avoided allergen on the first day, as well as on the second day of this two-bottle preference testing. And on the second day, this effect looks potentially slightly protective. And I think it will be interesting whether we can determine whether this is real or not by enhancing the power of mice and using more specific genetic tools in the future. But at least the vagotomy does not dramatically affect the ability of these mice to develop the avoidance. With this in mind, since we didn't find an effective TRPV1 ablation or subdiaphragmatic vagotomy on the ability of mice to change their preference for ova, we began to consider whether a humoral route might contribute. And our lab had previously done studies investigating the role of a TGF-beta cytokine called GDF15 in mediating tissue tolerance upon bacterial inflammation. And what had been found since then was that GDF15 was actually capable of driving conditioned flavor aversion to similar extents as the gold standards in the field, such as lithium chloride. And it's known that GDF15 mediates these effects through its actions on neurons in the area postrema, which is an area located in the hindbrain with a leaky blood-brain barrier such that substances in circulation can act on neurons there. We observed that GDF15 was significantly induced in sensitized and food allergen challenged mice relative to non-sensitized mice, and that the magnitude of this induction depended on IL-4 receptor alpha, IgE, and went away with mast cell depletion. Moreover, we found an effect of ALOX5 antagonism on the magnitude of this induction and a partial effect of cis LTR1 and LTR2 antagonism on this induction as well. And so this just tells us that GDF15 is induced, although not to an extremely large level. It is induced upon allergen challenge, and it seems to correlate with these various factors that we found were required for the behavior. What was interesting is that when we looked into the intestines of these mice to try to figure out where GDF15 is coming from, we did this via qPCR, and what I'm showing here is RNAscope, staining for EpCAM in white, FC epsilon R1 in green, and GDF15 in red, was that despite the vast majority of mast cells, which is this green signal being found in the proximal small intestine, and fewer being found in the colon, we found that the red GDF15 signal showed the opposite pattern with minimal expression in the proximal small intestine and robust expression in the colon. Moreover, we saw that within the colon, GDF15 primarily colocalized with EpCAM positive epithelial cells that kind of flanked the colonic crypt, as can be seen here, and that both the mast cell expansion as well as the GDF15 induction observed was completely IgE dependent. So this tells us that GDF15 is induced during food allergy, and potentially it's coming from colonic epithelial cells. However, this really doesn't get at whether this GDF15 means anything for this behavior. To test this, we sensitized mice, as we have throughout all the experiments I've shown you, and then 1 day prior to the 2-bottle preference test, treated them with anti-GDF15 antibody or isotype control antibody. This resulted in the anti-GDF15 treated group drinking far more OVA as compared to the isotype treated sensitized group. And this led to a sensitization-specific amelioration of the decrease in OVA preference seen due to allergy. And so, because this was a lot of information, just to put it all together, basically what we found is that allergic sensitization can induce changes in allergen preference before it results in all other pathophysiological symptoms that we normally think of and that are normally studied, such as diarrhea and anaphylaxis. We find that this avoidance behavior crucially relies on IgE, FC epsilon R1, and mast cells, and that mast cell-derived cystinyl-leukotriene production is likely responsible for this behavior in vivo. In terms of what is sensing, what mast cells are doing, I would say that this is still not very clear to us. We've tried to eliminate TRPV1 positive nociceptors and see no effect. Similarly, subdiaphragmatic vagotomy did not yield any significant effect on this avoidance behavior as well. One pathway that we identified that may be able to explain at least a part of this effect is that GDF15, which is produced in a manner dependent on IgE mast cells and leukotrienes, was required for at least a phase of this avoidance behavior. And so looking forward and trying to think about what all of this data means, to me, one of the most interesting aspects of this work is to think of how many people who have allergies actually have very mild symptoms that might manifest as just changes in preference for a given food, or for instance, abdominal pain and bloating, rather than what is normally clinically picked up, such as anaphylaxis or diarrhea. And I put this picture of a kind of smoothie bowl from a place on Yale's campus up here because my fiance has nut allergies, but she loves these smoothie bowls, so I often pick them up for her on the way home from lab. And the cashiers always ask me after I request the granola be nut-free whether this is due to a true allergy or a preference. And I think that our work kind of blurs the lines between what is truly a nut allergy, for instance, and what is simply a preference. So I thought that that was kind of funny. I also wanted to kind of put forth that this mechanism that we found may be more broadly applicable. It's not totally clear in what context leukotrienes might mediate aversive behaviors besides simply in allergies. Moreover, I think the diversity of how mast cells interact with neurons to alter GI physiology or physiology at large needs to be more extensively studied. And then obviously the big black box for this project, despite us learning a lot along the way, is what sensory neurons in downstream brain regions are required for this behavior to occur. I want to thank many people for their help on this project. Esther, who started this project and who's co-first author with me on this paper, who's a wonderful mentor, who has her own lab, as I said, at ASU, has been a huge help, obviously. Jamie Cullen, who's a research scientist who helped with the vast majority of experiments that I presented today, Ruslan, and then the Dietrich, Lieberliz, and Andrew Wang lab. And with that, I'll thank my lab one more time and open it up for questions from the audience.
Thank you very much, Nathaniel. That was a great presentation and a lot of data, a lot of work. And so please, anyone in the audience, if you guys have any question, please raise your hand and I will unmute you, or if you want to type your question in the chat, I will read it for you. In the meantime, I would like to ask, I have more questions, Nathaniel, than answers, I think. So do you think if that there are two particular questions that ILCs in the intestine could be interacting with mast cells, like, because we know that neuropeptides could be released by ILCs, I think, in the lung. So do you think that might be happening there?
Yeah, I think that's a super interesting point. So we haven't shown that this cis-LTR2, for instance, expressed by neurons specifically is required for this behavior. One population of neurons that have had a number of different papers published on them recently are these NMU expressing neurons that are located in the ileum and colon of mice. If you look, these NMU expressing neurons also express cis-LTR2. So it's definitely possible that these leukotrienes are mediating some effect on this behavior through interactions with other cells besides just traditional sensory neurons that we normally would think of. I think that's a great question and something that will need to be explored very in great detail in the future.
Okay, and I will say, I will ask if if microbiota could be, if you do this experiment without microbiota, does the animal still have avoidant behavior to the antigen?
So I wanted to do this experiment so bad, but it was a little bit tricky for a number of different reasons. So we don't have a facility here where we were able to test germ-free mice, for instance, on this behavior, and germ-free mice also will have a number of other abnormalities related to ENS development in addition to type 2 immunity. I did try to at least look at whether GDF15's induction depended on microbiota by treating with broad spectrum antibiotics, and I didn't see any major effect. So I would say I don't really know, but I think it's technically challenging, but interesting to do in the future. Yeah.
Thank you very much, Nathaniel. So I think we have a question from Nuri, so I will unmute you.
Thanks. Can you hear me now?
Yeah.
Okay, great talk, Nathaniel. I have a question about so I was thinking that when the mice are drinking the water, so do you know if this, in the end, they're also smelling it? And mice, I guess, are also very driven by smell. So I was wondering if you have any evidence or thought about if maybe it's a similar mechanism in the olfactory neurons or some in the olfactory epithelium that might be also associated just not only with the gut but also in the nose.
Yeah, for sure. I think that's an excellent point. So some of the original studies on this behavior, besides utilizing this two-bottle preference test paradigm, also utilized a model where OVA is nebulized. And so I think certainly mast cell degranulation in the nasal mucosa could lead to similar preferences, except obviously they would be mediated through separate neurons likely. In terms of how mice are realizing that it's the allergen itself that is making them sick, we tried to look into a role for taste sensation mediated by TRPM5, but that wasn't required. And I kind of do suspect, like you're saying, that the smell of the ova, these mice are able to pair with what mast cells are doing. So I think that's a really interesting point, and likely you're correct. Yeah.
So if I guess I will take my last question if there's no more question in the chat. So what are your thoughts on the difference between the two mice strains? So we know that it's just because of the amount of IgE or something else that we are not looking at. I'm thinking about there was a paper from the Diane Matthews group about Tregs in the intestine that change because between the mice strains. I was thinking that it's just because of the number of IgE that you could produce in one cell, in one mice versus the other, or other T cell component of immune cells there.
Yeah, so I didn't really focus on this very much today, just for the sake of time, but C57 Black 6 mice are extraordinarily resistant to the development of this avoidance behavior. I've done quite a substantial amount of work trying to figure out why that is, through various intercrosses. And it's kind of interesting. You can find that if you intercross the mice, it seems like the avoidance behavior is separate from the development of diarrhea. So mice can still develop diarrhea, but for instance, will not have the avoidance behavior. I don't think this also doesn't correlate with IgE levels or actually the number of mucosal mast cells in the tissue. So there's something else besides mast cell number and IgE that might explain this difference. I've even gone as far to give the same amount of leukotrienes intragastrically to both strains, and B6 mice still have an attenuated response compared to bulb C. So initially, I would have never expected that, but there's something fundamentally different about how allergens are sensed between the two strains. Could be Tregs, could be something else. I think it's a really important area to follow up.
Have you tried cross-fostering the mice?
I have not tried cross-fostering the mice, but that's an interesting point. It's a little bit complicated. So I think microbiota likely is non-contributory. People have done work on the differences in microbiota between the two strains. And although there are differences, it's actually determined by host genetics, such that if you cross-foster, whatever the genetics of the strain is actually what dictates the microbiota, not the fostering. But I think it's a very good point and something I've thought about a lot. Yeah.
Okay. So finally, a question from Nuriban. So he asked if there's any difference between sexes.
There is a difference between sexes. We find that the behavior is a lot stronger in female mice rather than male mice. And while we don't know exactly why that is, there is evidence that testosterone levels can impact IgE-mediated mast cell degranulation, and that might be just one facet of this. It's known, for instance, that in kind of more extreme food allergy paradigms, males are also protected as compared to females through a similar mechanism. Yeah, good question.
So yeah, Maura Ortiz asked, how do you describe the translational aspect of this research?
Yeah, so I think that the translational aspect is to try to make, basically the way that I see it is to really study how signals involved in inflammation in the gut are perceived by the nervous system. And that could contribute to a wide variety of symptoms such as abdominal pain, bloating, malaise that may not be specific for food allergies. This is an area that's extremely poorly studied, and there is some evidence that, for instance, post-infectious IBS involves the interaction between mast cells and neurons that lead to some of these more kind of sensory symptoms, I should say. Beyond that, that's one facet. Beyond that, I think that our finding of leukotrienes being involved in this behavior kind of raises a number of questions related to if they have roles outside of the behavior and for food allergy at large. These are things that we're actively following up right now. Great question, though.
So thank you very much, Nathaniel. Thank you, everyone, for joining. So I put it in the box chat, our webpage. If you want to join our Lifter, our Slack group, Facebook, Twitter, LinkedIn. We're constantly looking for people interested in hosting seminars. If you want to present your work in our one of our seminars, please let us know. And we are really looking for open science, open discussions. And thank you, Nathaniel, for this opportunity. Thank you, everyone,
Yeah,
and
thank
see
you.
you soon. Bye.
Thank you. Bye-bye.
Bye.